Okay, so the reading for today was mainly about getting more really more specific with our photosystems and specifically the light reactions. So I don't think that you'll get super specific about the steps on the first test, but we are going to go over all of the diagrams and look at more of the differences between the two processes that are noted in our unit guide. So you need to flip to 195 so that we can look at the graphs together. Once you do that, the first thing that comes up is linear electron flow, and that's basically the way in which our electrons and our energy flow through the two photosystems. And remember, which photosystem comes first in the light reactions? Photosystem number 2. It goes photosystem 1, photosystem 2. So let's look at the graph that is 10.14, and we see that photosystem 1, it is absorbing that photon, absorbing that light. It is bouncing around there just like the molecules, going right up to the chlorophyll A, and we are getting the same process that we discussed yesterday, okay? Now, we're getting a little more specific because we are talking about whenever that reaction takes place, not only is a photon being absorbed, but a water molecule as well. And this is really important because what can we get from a water molecule? H+. You have to remember that. That's actually how we get our oxygen out of it, because that hydrogen is pulled from our water molecule, sorry, I can't speak, and the oxygen that is left turns into O2, which is what we breathe. So you take that H plus off the water, that aids with the electron bumping up, and that also helps with a electrochemical gradient within the thylakoid membrane, which again is important because what does this use? Just like mitochondria, it uses an electron transport chain, okay? It uses it twice, actually. It uses this after the first photosystem system, between photosystem 2 and photosystem 1, and then it uses it again after photosystem 1, okay? Now let's talk about the two different products. After photosystem 1, when it goes down the first electron transport chain, it goes through a cytochrome complex and produces an ATP. And on the second electron transport chain, it goes through NADP plus reductase, and produces an NADPH. Remember, NADPH is for photosynthesis, NADH is for cellular respiration. Okay? And these are enzyme catalyzed. Okay, so that is linear electron flow. You have to remember that. Linear electron flow uses both photosystem 2 and photosystem 1. But what happens if we're using cyclic electron flow? Well, that is discussed on the next page, page 196 in figure 10.6. If you'll notice, we are only using photosystem 1, and what is happening is we are creating literally a cycle. The energy goes up into photosystem 1, goes through that primary electron acceptor, and instead of going through NADP plus reductase, it goes back through the cytochrome complex and produces an ATP. So instead of producing one ATP and one ADPH, we are just producing ATP over and over and over and over again. And it goes in a circle, hence cyclic. Okay? So let's differentiate. Linear electron flow uses both photosystem and produces O2, NADPH, and ATP. When we do cyclic, we are only producing ATP and we are only using photosystem number 1. Okay. And again, both use an electron transport chain. That's important to remember. Okay. And again, ATP and NADPH are both used in our light chemical reactions, and water is splitting so that the H pluses are given off for that electrochemical gradient, and O2 is given off through the stomata so that we can breathe it in. Okay, now let's talk specifically about our electron transport chain. It works pretty much exactly the same as the one for cellular respiration, except it's kind of flipped. If you will look on page 198, you'll notice that whenever, it's almost as if cellular respiration is flipped inside out, okay? The thylakoid, the outside of our thylakoid, is where the light is going in, and it goes through the thylakoid instead of in the matrix, where it goes into the matrix, it goes out of the matrix and back into it. We are going into the thylakoid and out of it. But remember, they use electron transport chains just like the mitochondrion. Linear electron flow versus cyclic electron flow, where linear electron flow is using both photosystems, and the different things it produces, where linear produces both ATP, NADPH, and O2, and cyclic only is producing ATP, and it is only using photosystem number 1. I would also remember that photosystem number 1 is P700. Check me on that one. I'm pretty sure it's P700. Okay. Yeah, P700. Okay, I'm not seeing anything other than making sure that you understand that that H plus gradient is being created, and that's an important part of that electron transport chain being active, which is how our cytochrome complex and our NADP plus reductase is used. I would remember those two things. So if you look back at page 195 in the graph 10.14, you need to remember that there's 2 electron transport chains in use with linear electron flow with our cytochrome complex and our NADP plus reductase, okay? I would remember just that part where the cytochrome produces ATP, the NADP plus reductase produces NADPH, and that the cytochrome complex is the only one that's being used during cyclic flow. That's it.